Aaron, are you gonna do an intro before we
No,
start
you
with
can. Oh, yeah, just to give
just
everyone
start
sort
at
of the,
just
yep,
start at 9 o'clock?
yep, I'll do
Okay,
that.
all right. I wasn't sure whether you wanted to tell people about raising hands or
Yep,
Q and
yep,
A or stuff like that.
yep. I'll do like 30 seconds of housekeeping stuff and then you can go. Go ahead.
Okay, perfect.
Okay.
Thank you.
Mhm. Okay, good morning, everybody. It's 9 a.m. We're going to get started in just a minute. I might give it another minute or so, but I just wanted to let everybody know who's on right now that I'd like to welcome Dr. Susan Arnold. She'll be introduced by Yoji Sumizu, who is the co-program leader for the immunology program at the Masonic Cancer Center. During the seminar or after the seminar, I'll leave that up to Dr. Arnold. If you have questions, you have two options. You can raise your hand and I will unmute your mic and you can talk, or you can put your questions into the Q&A module and the speaker and the host will address them that way. So go ahead.
All right, thank you, Aaron. Good morning, everybody. Welcome to today's Masonic Cancer Center seminar, and it's really my pleasure to introduce our speaker today, Dr. Susan Arnold. Dr. Arnold is an assistant professor in the Department of Veterinary Clinical Sciences here at the University of Minnesota and the College of Veterinary Medicine. She obtained her doctorate in veterinary medicine from the University of Wisconsin and then completed a small animal medicine and surgery rotating internship here at the U. And then she went on to complete a residency in small animal neurology and neurosurgery at the University of Georgia before returning back to the University of Minnesota. And Dr. Arnold's main research interest is in spontaneous brain tumors in dogs and really investigating novel treatments for canine brain tumors, as well as their potential application to human brain tumors. And I'm sure many of you are aware of a lot of the great work that's being done in this area of comparative oncology here in the Masonic Cancer Center, with much of it being done, for example, in collaboration with Dr. Liz Bluhar. And so We're looking forward to Dr. Arnold's talk, which today will be the canine connection and an update on the parallels between canine and human gliomas for mutual novel treatment exploration. Susan, thank you for being here today.
And thank you for that introduction, and good morning, everyone. I'm very excited to be speaking with all of you today. This is a relatively high altitude presentation just about some of the important kind of updates to the literature in terms of why, it's not just my opinion, but a validated opinion that the dog is such a wonderful model for studying glioma in people. So I don't need to go over this in much more detail. I just realized, though, that I don't know a lot of you, and a lot of you probably don't know who I am. And so just, to reiterate, yes, I've been here for about a year and a half now. I work very closely with Dr. Liz Pluhart and Mike Olin on the Canine Brain Tumor Clinical Trials program here. And then I have a faculty appointment here in the Department of Veterinary Clinical Sciences. I'm also a PhD candidate and within the Comparative and Molecular Biosciences department. I've been doing that since I got back here, and my mentors are Dr. Liz Pluhart and Dr. Mike Olin. And I'm going to be sharing some of my PhD research with you towards the end of this presentation. So what I aim to do today is I have got 3 phases to my discussion with all of you. And so phase 1 is going to be a quick summary on canine brain tumors, because I recognize that the majority of you are not veterinarians, and so some of this information is going to be relatively foreign to you. So I just want to get us all on the same page for discussing canine brain tumors just inherent to dogs themselves. From there, then I will characterize the canine model by providing updated diagnostic classification as well as parallels to human glioma. And then for phase 3, I will give you an update on our brain tumor clinical trial program, talking about essentially a summary of the clinical trials and showing you that this is really the perfect marriage of translational medicine in terms of an impact on both dogs and on people. And then I will introduce you to my PhD project, which I'm very excited about and I think has some tremendous translational impacts in the future as well. So go ahead and get started with a summary of canine brain tumors. So canine brain tumors, unfortunately, are relatively common, especially as dogs are living longer. And we have an estimated incidence of about 20 cases per 100,000 dogs per year across all the different forms of brain tumors that we see. We divide brain tumors in dogs based off of them either being primary or secondary. Secondary also is referred to as metastatic. And it's about 50-50 in terms of if they're primary or metastatic tumors. Within the category of primary brain tumors, broadly, we have meningiomas, gliomas, and then other tumors, which encompass a wide range of other tumors like pituitary macroadenomas, choroid plexus tumors, et cetera. But the vast majority of the tumors that we see fall into the category of either meningioma or gliomas. And of course, today, what I'll be speaking about primarily will be gliomas, which in dogs are conventionally referred to as astrocytomas, oligodendrogliomas, and then unspecified tumors. Gliomas are reported to make up about 33% of all primary brain tumors, and of those, 66% of gliomas in dogs are oligodendrogliomas. Now, I have an asterisk here because that's what's reported in the literature, but I will say that clinically, I think that gliomas make up a larger proportion of primary brain tumors than 33%. Historically, it's been the case that meningiomas have been the most common brain tumors in dogs, but I think that as we've seen a rise in the popularity of brachycephalic breeds, that distribution is becoming more skewed towards a higher prevalence of oligodendrogliomas. When we look at gliomas, and specifically oligodendrogliomas for that matter, the patient profiles of these dogs are that they tend to be middle-aged to geriatric dogs. The average age at the time of diagnosis is between 7 to 8 years old. We've seen them in as old of dogs as 14, seen them in as young of dogs as 3, but in general, somewhere around 7 to 8 is kind of the unfortunate kind of magic number for these dogs for when they develop their gliomas. There's a slightly higher prevalence of gliomas in male dogs compared to female dogs, but this is, I would say, just very, very slight at this point. What is definitely observed is a risk factor associated with the breed of dog, and specifically kind of the overall physical conformation of the dog. So brachycephalic dogs are dogs that are characterized by having these short little snub noses. And the most common dogs that we see gliomas in that are brachycephalic are here, okay? And so these are boxers, Boston Terriers, French Bulldogs, and Bulldogs in general. And these short-nosed dogs are those that are just classically considered to be overrepresented in developing gliomas, and especially in developing oligodendrogliomas. The tumor locations are similar to those that are observed in people. And so the majority of dogs will develop their gliomas in their frontal, parietal, and piriform lobes. They will also develop caudal fossa tumors, meaning affecting the brainstem, but it's generally much less common than to see them in the prosencephalon, or the big meaty part of the brain. What we observe in patients with prosencephalic tumors is the vast majority of these dogs will have seizures as their only clinical sign. They're neurologically normal. We can't ask them if they're having a headache or anything like that. And so overtly, they seem relatively normal, aside from an acute onset of seizures at a later age. If the tumor is allowed to progress, or in the more advanced stages of the disease, as the tumor kind of reaches a critical volume, they'll start to develop neurological signs with it. And so the things that we see in these patients are obtundation, a propulsive gait, and a contralateral hemi neglect. To be obtunded means to have a decreased responsiveness to external stimuli. And so these dogs just kind of look like they're in a fog. The propulsive gait that we see is classic for a prosencephalic lesion. These patients have a normal gait in the sense of they have no weakness and they have no ataxia. So no paresis and no ataxia. So they walk normally, but when they walk, it almost looks like they feel compelled to keep walking. It's like an obsession that they have, and they will circle in the direction of their lesion. And so owners will report, oh, he just like constantly circles around the dining room table at home, and he's always circling to the right. So circling towards his lesion. The other thing that we see in these patients is a contralateral hemi neglect, meaning that on the side opposite of their lesion, they will have neurological deficits in their processing of the information coming in from that side. So they'll have something like a menace deficit, which is if we make a menacing gesture towards their face, they should protect their eyes and blink. And so they won't do that. They'll also have a nasal hip algesia, meaning that if you stick something noxious into their nasal cavity, they should try to get away from it. They won't do that if they have this contralateral hemi neglect. And they'll also have posture erection deficits, which is because they have abnormal perception of the awareness of their limbs in space. They have decreased kinesthesia, and they will also have so they'll be contralateral to their lesion for the same reason. So this is a video of a patient with a prosencephalic lesion. And so what you can see in this video is that he's got a relatively normal gait. But as he walks around the room, at every chance that he has to either go left or right, he always goes to the right. I joke with students, and it's becoming less and less relevant because fewer of them have seen the movie Zoolander, but the quote of, "I'm not an ambi-turner, I can't turn left," that's what these dogs do. I don't get a laugh as much anymore because fewer of them have seen the movie, but for a while it was really funny. And so essentially he's got a very normal gait, but as he's walking around, like I said, at every opportunity he turns right. I'm just gonna fast forward through this because you don't need to keep watching a dog just walking around. And then get to the other parts of his exam that you'll see are abnormal. So this is our posture reaction testing. So on the right side, he's pretty normal. So he catches himself before he would fall. On the left side, he has a delayed reaction. So his foot crosses midline before he corrects himself. And so that's a posture reaction deficit. So he's got contralateral left-sided deficits referable to his right prosencephalic lesion. And yes, that is my colleague in a boot. She wore that for the better part of a year. And we joked that in all of our videos, we've got her in a boot. She's just that tough. But so once again, right pelvic limb, relatively normal. And then on the left side, not so great. And then normal menace on the right and no menace on the left. So once again, consistent with the right prosencephalic lesion. And then this is testing for his nasal hip algesia. And you can see that when he has the hemostats pushed on the right side, he reacts. But on the left side, he doesn't do anything. So that's his nasal hip algesia. And so the reason I want to show this to you is because I want to demonstrate that it's relatively easy to monitor the neurological signs in a dog. And so, compared to something like a mouse, it's a lot easier to do a neuroexam in these patients. And so we have relatively tractable clinical signs that we can monitor over time, which just adds to the benefit of the dog as a model for glioma. And then this is just an overall schematic about what we're dealing with in terms of what these tumors look like. So on the top, we've got a canine oligodendroglioma. Bottom is a canine astrocytoma. What you can see is that on an MRI, this is a T2 weighted transverse of an oligodendroglioma. These are interparenchymal masses. They're highly T2 hyperintense due to the mucinous component of these oligodendrogliomas. And then over here, just show you kind of the classic kind of fried egg appearance that occurs due to fixation artifact in these tumors. So this is a classic example of an oligodendroglioma. Canine astrocytoma can have variable appearance on MRI. This one is relatively, obviously, very T2 hyperintense, and so it has a relatively high water component to it. And then once again, just shows that it is an interparenchymal mass that distorts the surrounding parenchyma. And here's just a relative example of what histopathology looks like for a canine astrocytoma. The devastating thing about brain tumors in dogs is kind of twofold. One, it's just a very devastating disease in general, just like it is in people. And two, we still just really don't know a lot about how to best treat these animals. And so broadly, there are 3 different approaches for treating brain tumors in dogs. We've got palliative care, which consists of just keeping them comfortable, reducing the tumor side effects with anti-inflammatory doses of steroids, as well as anticonvulsants if necessary. And then for definitive care, we've got surgical resection and radiation therapy. Chemotherapy is really not a beneficial endeavor for canine brain tumors in general, because most of the drugs just don't get past the blood-brain barrier effectively. So temozolomide, for example, I know it's standard for use in glioblastoma in people, but it just has not been demonstrated to be useful for dog gliomas because of the blood-brain barrier. When we look at the survival time by treatment type, if you look at the literature, there's a pretty decent retrospective study that came out in, I think, 2015 that looked at the survival time based off of dogs that received palliative care alone. And what they found was that across all locations of tumors in the brain, median survival time was about 70 days. Comparatively, if they just separated out and looked at it by location, dogs that had tumors rostral to the tentorium cerebelli, meaning supratentorial, had a survival time of 178 days, as opposed to dogs where it was caudal to that with a survival time of 28 days. What they found with histopathologic confirmation is that for palliative care, the tumor type does not matter in terms of survival time. What matters is the location of the tumors. For surgical resection, challenge here is that we have limited kind of histopathologic confirmation. There's a systematic review that came out in 2015 that looked at overall survival time for surgical resection. And they found that for all tumor types and locations, the median survival time was 312 days across all tumor types. There's a subsequent paper that came out in 2019 that showed that for dogs that had gliomas, histopathologically confirmed gliomas, who received surgery alone, the survival time was 66 days. So a marked difference for glioma patients compared to all tumor types. And so I think this is a more reliable number to provide to clients based off of the fact that in this second paper, there was histopathologic confirmation of the tumor type. Radiation therapy is also kind of a mixed bag for all tumor types and locations, and for all radiation therapy protocols, the median survival time is about 351 days, according to that systematic review. But the glioma response specifically is unknown. And so the take-home points here are that we really have limited treatment options for dogs with gliomas, and we don't really know the glioma-specific survival information. And so we can take that challenge and turn it into an opportunity. And so overtly, if we look at the dog, we can just kind of off the cuff think of a number of benefits for using dogs as a model for glioma in people. For one thing, these are spontaneously generated tumors, and so that means that these tumors co-evolve with the tumor microenvironment co-evolving with their overall microenvironment, right? They have intertumoral heterogeneity, just like we see in human glioblastoma. The blood-brain barrier is intact, which is a critically important thing to be evaluating in patients who would also have an intact blood-brain barrier on the human side. And also the intact immune system is critically important and something that makes the dog a superior model to the mouse model, right, which doesn't have an intact immune system when we have these xenografts. And the intact immune system is critically important because we know that there's so much interaction between the immune system and gliomagenesis and glioblastoma propagation. And so we can take those benefits and take those take-home points in the fact that we have limited treatment options for the dog and we have glioma-specific survival kind of unknown information, and we can turn that into using the dog as kind of the missing puzzle piece for determining how best to treat canine gliomas and also human gliomas. And so that's going to require us to discuss characterization of the canine model. And so I'm going to go on now to phase 2 of this discussion. And what I'll do is I'm going to divide this part into 3 kind of subsections. And so first, what I'll do is provide you with the revised diagnostic classification of canine brain tumors. And then I'll show you the genetic and molecular landscapes of canine brain tumors relative to human brain tumors, both for pediatric and adult gliomas. And then from there, I'll show you also how the systemic and local immune profiles of canine gliomas are similar to human gliomas, all of which I hope will really emphasize why the dog represents a really reputable model for studying glioma, both in adult and pediatric glioma. So this is the algorithm that was developed for classification of diffuse gliomas based off of histological and genetic features. And so this is out of the 2016 WHO classification of tumors of the central nervous system. I'm sure for those of you who work with glioma and work with brain tumors, this is all very routine at this point for you. But I think that there are probably a number of people who are not familiar with this algorithm. And so I'm just going to kind of go over it in general. And so historically, the last update that came out from the WHO was from 2007, and it classified gliomas like every other classification system, which was based off of microscopy identification, so histopathologic diagnosis exclusively. And in 2014, there was a group of people who met to essentially kind of start to lay the groundwork for including genetic markers in brain tumor diagnosis. And so some of these canonical pathways were known at the time of the 2007 WHO guidelines, but they just weren't thought to be validated enough to include in that WHO guidelines. But they've now been incorporated into the 2016 WHO classification. So this is the algorithm that came out of that classification scheme. And what you can see here is that tumor diagnosis is now based off of both including a histopathological name as well as genetic features. And when there's a discrepancy between the histopathologic diagnosis and the genetic, the genotyping, I should say, the genotyping wins at this point. And so when we look at glioblastoma, they're characterized as either being IDH mutant or IDH wild type. IDH stands for isocitrate dehydrogenase. And so if you were to be diagnosing a patient with glioblastoma, you would identify them as either being glioblastoma, IDH mutant, or glioblastoma, IDH wild type. And you can see here that this is a comparison between the 2007 scheme and the 2016 scheme. And so now when we look at glioblastoma, which is WHO grade of 4, those are now characterized as one of 3 things, either the IDH wild type, the IDH mutant, or NOS, meaning that not enough information was available at the time of diagnosis to determine the IDH status of the patient. And this overall new scheme completely breaks a century-old technique, which was historically based exclusively on histopathology. So incorporating this genetic information was really groundbreaking and clearly very important. And what I wanted to show you here is the importance of this revised diagnostic classification in people. And so this isocitrate dehydrogenase, excuse me, has really important prognostic implications. Again, this is probably very obvious to those of you who do glioblastoma research, but for those of you who are unaware of this, just wanted to bring this into light because it really validates the methods of using genetic profiles of tumors to determine the grade of them. And so isocitrate dehydrogenase 1 and 2 are key Krebs cycle enzymes that are NADP dependent. And what they do is they catalyze the oxo-decarboxylation of isocitrate into alpha-ketoglutarate. And this has protection from oxidative stress in the cells via kind of interactions with the glutathione and thyroreduction producing pathways. And IDH1 and IDH2 are also involved in glutamine metabolism under hypoxia and also in the electron transport chain alterations. These mutations in isocitrate dehydrogenase occur early in the pathogenesis of gliomas. All of the mutations of IDH in people are monoallelic, and they're mostly missense variants that lead to single amino acid substitution of arginine residues at codon 132 of exon 4 for the IDH1 gene, or codons 140 or 172 of the IDH2 gene. And these mutations are not only loss of function mutations, but also lead to a gain of function mutation in the formation of 2-hydroxyglutarate, which is an oncometabolite. This little schematic here just kind of shows you the ramifications of IDH mutations in cells, and ultimately what it leads to is mutagenesis and tumorigenesis. And then this is a subset from the summary of the WHO guidelines, which shows you just how important this isocitrate dehydrogenase marker is, in that it really delineates survival time. And so patients who are the wild type have a significantly shorter survival time compared to the IDH mutants. It's not really clear why the IDH mutants do better with therapy compared to IDH wild types. But what is clear is that this is a very important delineator between these two subtypes. And then you can also see, based off of what I have here, that what this also delineates is differences in mutations of other really key oncogenic promoters in these glioblastoma patients. And so hopefully just what this shows you is that this is just validation of using these genetic markers for classifying brain tumors. And this is something that we haven't really done yet extensively in dogs, and so we need to start doing that. And so to that effect, where we're at with canine classification of oligodendrogliomas and other canine gliomas is that the National Cancer Institute Comparative Brain Tumor Consortium developed a glioma pathology board, which consists of a combination of veterinary and physician neuropathologists. And what they did was they sat down to do a comprehensive review of 193 cases of canine glioma. And the whole goal of this consortium pathology board was that they wanted to really lay the groundwork for modeling the new WHO 2016 guidelines so that they could have a very parallel histopathologic investigation of canine brain tumors, which would be simplifying and would unite the histologic classification, and then would also allow for kind of nestling and incorporation of a panel of key biomarkers in the future. And so this is a representation of their decision tree, and so a much simplified way of going about diagnosing brain tumors in dogs. And so what they do first is they characterize morphology as either an astrocytoma, an oligodendroglioma, or unspecified or undetermined. They then look at the infiltration of the tumor. And so what they're looking at is if there's no infiltration, that means that's an N. F stands for focal, and D stands for diffuse. So they go on then to look and see if there's necrosis and or microvascular proliferation and pseudopalisading. If those features are present, then the tumor is considered high grade. If those things are not present, then they go on to look for mitosis as well as overt presence of universal features of malignancy. If those are present, then the tumor is considered high grade. And so the whole purpose of this was really to mirror what the WHO guidelines do so that in the future, we can incorporate genetic biomarkers in to allow for a unified classification of canine brain tumors relative to human brain tumors. These are some representative images from that atlas that they developed in that pathology board. And so these are examples of what a canine high-grade oligodendroglioma looks like. And so what the image on the left is showing is the microvascular proliferation. So you can see these tortuous vessels, they're thick. They've got a lot of endothelial cells surrounding them as well as parasites. And so increased microvascular proliferation is a key hallmark feature of high-grade oligodendrogliomas in dogs. And then this one here, what I'm showing you is the area of necrosis. The arrow here is pointing to some pseudopalisading, which may or may not be accompanying the necrosis that we see. What's important is that the degree of invasiveness in high-grade oligodendrogliomas in dogs is not associated with the grade. And so this tumor, you can see, has a lot of necrosis in it. And I know it's obviously hard to see, but these are areas of microvascular proliferation. But what's different is that there just isn't a lot of invasiveness. And so even though this is a high-grade glioma, it's not leading to invasion of the subjacent white matter here. Looking at further into the classification, when we look at immunohistochemistry, of course, OLIG2 is going to stain very strongly for oligodendrogliomas. Can have a bit of staining for astrocytomas in dogs as well, although the staining pattern is just not going to be as robust as it will be with an oligodendroglioma. OLIG2 is relatively resistant to fixation artifacts, but some of the other immunohistochemical stains that we use are not as resistant. And so one of those is CNPase. And CNPase is great for diagnosing oligodendrogliomas because it's specific to oligodendrocytes and Schwann cells. And so it will help with distinguishing between oligodendroglioma and astrocytoma line for tumors. But it is a little bit more sensitive to artifacts from fixation. And so overall, if we don't see this, it doesn't necessarily mean that we should not diagnose a patient with an oligodendroglioma, especially for necropsy tissues where those tend to just be more affected by the artifacts. So now what I want to do is now that I've kind of given you just a running kind of impression of what canine oligodendrogliomas in particular look like, I want to now characterize the genetic and molecular landscape of these tumors relative to human glioma. So this figure comes out of a paper that was published in Cancer Cell in 2020. And what this group sought to do was they wanted to compare the molecular life histories of spontaneous canine and human gliomas. And they looked at both adult and pediatric human gliomas compared to canine gliomas and did comparative genomic, transcriptomic, and epigenetic profiling. And so they used the updated diagnostic classification that we just discussed to diagnose dogs with oligodendrogliomas, astrocytomas, or unspecified. And what this figure shows you is that essentially what they did was they grouped genes by what they do, right? And so deregulating cellular energy pathways, genome instability, for example. And then they compared the proportion of patients that show alterations in these driver events between species and within the different tumor types for people. And so CG stands for canine glioma, PG stands for pediatric glioma, AG stands for adult glioma, and then these are the WHO subtypes for each of those. And what they found was that the proportion of gliomas with at least one significantly mutated gene was about 44% in dogs. 52% of pediatric gliomas had alteration of at least one significantly mutated gene, whereas 93% of adult gliomas had at least one significantly mutated gene. And so the conclusion is that there are similar mutation rates in canine and pediatric glioma. And this is emerging information that is really important. And it's something that I think has been maybe underutilized at this point and under-investigated. But this is really exciting to me because I think that, finding that we have, a model potentially for pediatric glioma in addition to human adult gliomas is really exciting and something that's kind of groundbreaking at this point for these dogs. So they then went on to do further work to kind of drive home the point of the similarities between canine and pediatric gliomas. And so when they look at the subtyping, right, with adult gliomas, that's the IDH mutation. For pediatric glioma subtyping, based off of the WHO guidelines, they're subtyped based off of histone 3 mutation, so either wild type or the mutant form of it. And what they found was ultimately not many of the dogs had IDH mutations. I think there were only 3 samples that had an IDH mutation out of the 83 dogs that they profiled. So not all that common to see that mutation. What they found, though, was that canine gliomas were most similar to pediatric gliomas in a number of other ways. So one of the things that they found was that they were most similar to the H3 wild type pediatric gliomas at the pathway alteration level. They also found similar mutation rates to both the H3 wild type and mutants, which is what I showed on the previous slide. And then they also found similar rates of aneuploidy. And in dogs and in the H3 mutant, the rate of aneuploidy, which is shown here, is 25 and 26% respectively, which is much higher than any of the other groups. When they compare methylation profiles between dogs based off to kind of an established guideline in people, they found that 78% of the canine tumors were classified as pediatric based off of their methylation profiles. So once again, just really driving home the point that dogs represent an emerging model for studying pediatric gliomas as well. So now what I'm going to do is I'm going to focus specifically on oligodendrogliomas and canine oligodendrogliomas, I should say. And that's because there is now emerging evidence that canine oligodendrogliomas are more similar to human glioblastomas than they are to oligodendrogliomas. And I'm just going to show you some evidence to support this claim. And so in this paper, they compared canine oligodendrogliomas from brachycephalic breeds to human pathways. And this is pretty important because I think it's really important that we specifically investigate oligodendrogliomas in brachycephalic breeds because it's a smaller patient population, and so we're more likely to catch conserved mutations in a smaller genetic pool than if we're looking at oligodendrogliomas across the board, especially also because the oligodendrogliomas in other breeds might be different than the oligodendrogliomas in this shared group of breeds. And so what the figure on the left shows is gene expression in normal and canine glioma brain tissue for a set of genes that are associated with human glioma. And so, for example, you can see here that TP53, what they found is in the glioma tissue, this was highly expressed compared to the normal tissue. And this is important because 87% of GBM mutations have some sort of a mutation in this pathway. When they then did gene ontology and KEGG pathway enrichment, what they found on these cluster differentially expressed genes, when they characterized them, was that upregulated pathways included those involved in transcriptional activity enhancement, cell cycle progression, et cetera. And similar to human brain tumor pathway downregulation, they also found that those that were downregulated were things involved in neuronal maintenance. And so this overall kind of pathway pattern of what's upregulated and downregulated is very similar between canine oligodendrogliomas and human glioma. Here's another study that shows similar conclusions. And so in this paper, they evaluated the overall gene expression landscape of canine oligodendrogliomas and found a total of about 3,700 differentially expressed genes that were either upregulated or downregulated with a log 2-fold change that was greater than 2 in canine tumors compared to normal brains. Then what they did was they took the top 10 upregulated genes, which are shown here in blue, and they compared them to human genomic databases. And what they found was that when they compared these upregulated genes from the dogs to the human genomic databases, they found that 80% of the upregulated canine genes were also upregulated in human oligodendrogliomas, but importantly, 90% of the upregulated canine genes were upregulated in glioblastoma multiforme. And so this suggests that genetically, canine oligodendrogliomas behave more similarly to human glioblastoma than they do to human oligodendrogliomas. And I think that this is really relevant because glioblastoma multiforme is, of course, the most severe form of human brain cancer. And so now we have the most common form of canine brain tumors being oligodendrogliomas, showing that they are more similar to the most severe form of brain tumors in people. And so we have a relatively abundant canine model for a very severe human disease. So going off of that, not only are they genetically similar, but canine oligodendrogliomas also share immune profile similarities with human glioblastoma. And so what I'm showing you here is evidence for immune cell infiltration profile similarities between oligodendrogliomas relative to people. And so what this is showing here is compared to normal tissue, what you can see in canine oligodendroglioma tumors is that there's an increased prevalence of percentage, I should say, of CD8 positive T cells, as well as regulatory T cells here. And then likewise, when we look at canine peripheral blood, what we can see is that there's an elevation really similar to what they see in people. And so we see an elevation of regulatory T cells in circulation as well as elevations in CD8 positive T cells. And so this mirrors what's seen in people with their systemic and local immune profiles for people with glioblastoma multiforme. And what this also suggests is that similar to in people who have circulating elevated levels of regulatory T cells, suggests that these regulatory T cells in dogs are also important in the immune suppression features of gliomogenesis in dogs. So once again, similar profiles between canine oligodendrogliomas and human glioblastoma. This is further evidence that canine oligodendroglioma is more closely something that resembles human glioblastoma rather than oligodendroglioma. And so these are the top 10 cluster differentiation genes that are upregulated in canine oligodendroglioma. Only 11% of them are upregulated in human oligodendrogliomas, as opposed to 80% of them are upregulated in glioblastoma in people. So once again, very similar profile relative to human glioblastoma for these canine oligodendrogliomas. So if we turn our attention to immune checkpoints, what we can see is that there's evidence for PD-1 and CTLA-4 expression in canine oligodendrogliomas. So what they're showing here is that there's prominent PD-L1 staining for canine glioma tissue. CTLA-4 was also detected. And just for comparison, 88% of human glioblastoma expresses PD-L1. So again, very similar to what's seen in the dogs. And then likewise in peripheral blood, compared to healthy controls, there's a significantly greater PD-1 expression on CD4 positive and CD8 positive T cells from glioma bearing dogs. So once again, pretty similar profile to what's seen in people. Another important immune checkpoint that I want to talk about, because this is the focus of my mentor's research, is the CD200 immune checkpoint system. So CD200 is a highly immunosuppressive protein that's secreted by tumor cells and is also upregulated on tumor vascular endothelial cells. And this immune checkpoint pathway is regulated through 2 different receptors on antigen presenting cells. And so we have inhibitory receptors, which are the default receptors of the CD200 peptide. And then there's activation receptors, which normally the CD200 doesn't bind to. And when CD200, when it's cleaved by matrix metalloproteinases and is circulating, when it binds to antigen-presenting cells, it suppresses the secretion of pro-inflammatory cytokines and also increases myeloid-derived suppressor cells and regulatory T cells, and overall compromises the anti-tumor response. And so this pathway significantly contributes to immune suppression in glioblastoma. So Dr. Olin and his colleagues created a CD200 peptide, which is CD200 activation receptor ligand. And what it does is it binds to the activation receptor on these antigen presenting cells. And in doing so, it allows for bypassing of this pathway that normally occurs when CD200 binds to the inhibitory receptor. And in doing so, it upregulates CD80-86 expression and also facilitates antigen presenting cell differentiation into dendritic cells. And then it downregulates inhibitory checkpoints like the PD-1, PD-L1 pathway, as well as CTLA4, and of course, the CD200R1 pathway as well. And in doing so, this allows immune cells to move across the immunological barricade. And this is important because in human GBM, increased CD200 receptor expression has been significantly associated with decreased survival times. And so what I'm showing you here is a Kaplan-Meier curve from our canine patients that are enrolled in our ongoing clinical trials. And so what you can see here is that compared to dogs that received an autologous tumor lysate vaccine alone, dogs that also received the peptide, the activation receptor ligand, have a significant survival benefit over dogs that didn't receive it. And so essentially doubled the survival time for patients that received the CD200 ARL compared to dogs that didn't receive it. And then at the same time, what you can see here is that as we track their progress, these are months. And so 0 months, 4 months, 8 months, and 12 months. And what you can see this is flow cytometry from peripheral blood mononuclear cells, is that we see a decrease in the CD4-CD8 ratios here. And then we also see a decrease in the expression of PD-1 for both CD8 positive and CD4 positive T cells. There's also a decrease in myeloid-derived suppressor cells over time, as well as an increase in the CD8 granzyme B population. So overall showing a favorable response to this therapy. And this is exciting because it has paved the way for what's now an ongoing phase 1 clinical trial going on here with people. And so kind of to summarize this part of this, what I want to say is that compared to human GBM, I think when we're thinking about whether or not canine oligodendrogliomas are a good model, we should ask ourselves, do they share similar life histories? And absolutely, that's true. They share similar life histories. They have a relatively complicated sharing of their genetic profiles and molecular profiles. I would say for the most part, what we have found is that they certainly share a lot of similarities in their genetic and molecular profiles. One key difference that is still kind of up for debate is the IDH pathway or mutation in dogs relative to people. And there's kind of varying reports right now on that. Kind of my naive opinion about this is that because the IDH wild type people are the worst of the worst in terms of their survival rates, I'm not sure that it really is that important that dogs would be identified as having the mutant form, because I think it's more important that we are working with wild type patients because they will more similarly mirror the mutation profile in people with GBM, and in particular, people who are IDH wild type. And then they certainly do share similar tumor microenvironments, as I showed you with the immune profiles. And so briefly, what I'd like to do now is just kind of bring you all into the fold of what's currently happening with our canine brain tumor clinical trial program here. And so this clinical trial program has been established here for over 12 years. It's a highly successful program that I have just kind of walked into in the past year and a half. And so I can't have any credit for what's been done here, but incredibly wonderful work between a variety of departments here within the Veterinary Clinical Sciences Department, Pediatrics, and Neurosurgery. And then this brain tumor clinical trial program is nestled within a larger program itself, which is PRECINCT, which is the Pre-Medical Cancer Immunotherapy Network for Canine Trials, which is supported by an NCI U01 grant, as well as a Cancer Moonshot and Immuno-Oncology Translational Network. And the goal of PRECINCT is to launch translational projects that will accelerate immunotherapies for people. And for our clinical trials, the enrollment criteria are essentially we take adult dogs with MRI-confirmed intraparenchymal masses that look like gliomas. We have excluded French Bulldogs, and I'll get to that in a second. The general scheme for what happens with these patients is they come to the University of Minnesota, they undergo a decompressive craniotomy, and we do a histopathologic diagnosis as well as make autologous tumor lysate vaccines for those dogs that are enrolled in that branch of the trial. They then go on to have their immunotherapy treatment. And this is kind of a calendar here to show you just the general approach. It's a year-long enrollment. And so they have surgery and then they have an MRI every 4 months for a year. They also have peripheral blood characterization via flow cytometry on their PBMCs every 4 months to just kind of see what their immune profiles are doing over time. These red arrows represent times when they are receiving their immunotherapy treatments. And so we track these dogs for a year or until they have evidence of tumor regrowth on their MRIs. The current trials that we have going on right now, this is just a subset of them, but we have 3 kind of main branches. We have dogs that receive an autologous tumor lysate vaccine with the CD200 checkpoint inhibitor. We also have a gene therapy group where they receive either the gene therapy alone or with the CD200 checkpoint inhibitor. Overall, I would say it's going very well. We're still in our recruitment phase. And so kind of stay tuned from kind of my mentors for further information about where this is going. But overall, I would say it's going very well. So I want to just kind of harp on this French Bulldog exclusion really quickly and introduce you to my research. And so the reason that we've excluded these French Bulldogs is because uniformly, when they were enrolled in the trial, they experienced a poor response to immunotherapy. They neurologically deteriorated really quickly, they had severe seizures, and they had rapid tumor regrowth. And their median survival time was 48 days. And I'll remind you that that's worse than surgery alone, right? Surgery alone for gliomas in that paper from 2019 described a median survival time of 66 days. So median survival time of 48 days in these patients is obviously devastating for us and very different from the rest of the dogs. And so this is just kind of an example of overall non-French Bulldogs and the percent survival in our immunotherapy trials. And comparatively, you can just see that the French Bulldogs have just done really terribly. So there's something very different about this distinct breed, and so essentially this is my thesis for my PhD. And what I'm hoping to do is I want to elucidate the mechanisms that are responsible for the poor outcome of this breed when they're treated with immunotherapy. And questions I have are, is this because of differences in the tumor? Is it because of their immune system being different? Is it a combination of tumor, immune, and immunotherapy interactions? Does it have something to do with the fact that these are short-nosed dogs that are extremely brachycephalic? Do they have chronic hypoxia contributing? So essentially, what's the kind of the interplay between all of these potential factors and others? And so I think that overall, these differences contribute to a uniformly poor outcome in the French Bulldogs compared to other breeds where HGG stands for high-grade glioma. And so what I'm planning on doing is I'm going to first look at tumor differences by using stored tumor tissue pre and post treatment from French Bulldogs and Boxers. And Boxers represent a good controlled population for us. And so we'll characterize the tumors with gene expression analysis and copy number variant analysis, and then look at the immune microenvironment via in situ hybridization assays. And these are just kind of my control or my comparison groups. I'll then kind of focus on immune differences. And so I'll look at the peripheral blood mononuclear cell profiles between French Bulldogs and Boxers using nanostring to look at T-cell analysis, et cetera. I anticipate that given that there's a lack of genetic diversity in dog breeds, that there's a high likelihood of shared tumor mutations in these French Bulldogs. And I also think that because immunodeficiencies are inherited in other dog breeds, that we might find that there's some sort of an immunodeficiency in these French Bulldogs as well, which alters their immune response in response to the tumors and in response to immunotherapy. And what I'm ultimately hoping to do is investigate a correlation in people, because as I've hopefully shown you now, there's a high similarity between human glioblastoma and oligodendrogliomas. And so if we find conserved mutations in the French Bulldogs, we might be able to find that there's kind of a patient population who would potentially respond poorly to immunotherapy, and we can start to direct targeted therapy towards that group. So overall, in conclusion, what I'd like to just kind of say overall is that hopefully what I've driven home is that canine gliomas are an excellent preclinical model for human gliomas, that there's growing evidence for canine gliomas as a model for pediatric gliomas, and there's growing evidence for canine oligodendrogliomas as a model for GVM. and that ultimately investing the poor outcomes in French Bulldogs might have some translational impacts in the future. I'm of course very excited about all of this. I'm really excited about the pediatric glioma part of this because I think that this is represents another tremendous opportunity for using dogs to study cancer in people, and pediatric gliomas are certainly one that pulls at the heartstrings. So I would like to thank you very much for your attention today, and I would especially like to thank my mentors, Dr. Pluhar and Dr. Olin. I'd also like to thank my research sponsors. And so my research is funded by the Daniel and Sheila Sack-led Brain Tumor Research Grant through the Children's Cancer Research Fund. I am immensely grateful for this funding, especially because it's going to allow me to investigate the interactions or the relationship between canine oligodendrogliomas gliomas and pediatric gliomas. And so I'm exceptionally grateful for that funding. I'm also thankful for my funding through Grant and Aid, which is allowing me to get my foot in the door as a junior researcher, and then also to the American Kennel Club Canine Health Foundation. So thank you very much for hanging in there with me. This was, I know, a whirlwind, and I'm happy to take any questions at this point. Should I maybe stop sharing my screen at this point? Would that be helpful?
Sure, Susan, I think that would be helpful. Thank you.
Okay.
Thank you very much.
Yeah, my pleasure.
And if there are questions, you can write them in the Q&A or raise your hand and we will unmute your microphone and you can ask the question or put it in the chat as well. Aaron, it looks like Dr. Yee has a question.
Yep. Let me allow to talk and go ahead, Dr. Yee.
Hi, Dr. Yee.
Hi, thanks. That was a really great presentation, showed the power of comparative oncology. I'm wondering if you had any thoughts about germline predisposition, in particular, brachycephalic dogs. Are there germline observations that can be made that relate to risk of developing brain tumors?
Yes, that is,
things that are not genomic changes in the tumor.
Yes, excellent question. So there are some, and I'm sorry that I didn't address that, but yes, there's certainly evidence on the canine side that a lot of these tumors have a profound germline kind of driving effect. And I think that you're right that investigating that with the brachycephalic dogs, given that this is maybe a co-developing condition, would be an excellent thing to investigate in those dogs. And I thank you for that.
Okay, thanks.
Yeah.
There's a question in the chat about the blood-brain barrier and whether there were significant differences in dogs compared to humans.
Are there significant differences in the blood-brain barrier between dogs and humans? I would say there must be some, because yes and no. So there's difference in the I would say probably in the drug efflux systems. I don't know a lot about the specific molecular features of the human blood-brain barrier, but what I do know about dog blood-brain barriers is that there's a lot of drug efflux systems in the dog blood-brain barrier, which is responsible for basically pumping out over 70% of drugs that would otherwise get into the canine brain. And so temozolomide is one of those that probably is just kicked right out because of that efflux system.
All right, great. There's another question here from Sarah. So you showed some of the shared genes that are expressed between the human and the canine gliomas, and some of the different categories like transcriptional activity enhancement and cell cycle progression, angiogenesis, et cetera. Have you looked further into the DEG analysis and coupled it with ChIP-seq data or some other tool to try and get at whether there are specific transcription factors that are mitigating the expression of these genes?
That is a great question. I personally have not done that. I do believe that as part of their analysis, they did some of that and were able to kind of confirm kind of at a transcription level what's driving some of these. I can get you more specifics and I'm happy to share the paper with you, just kind of go over everything. But yeah, that was certainly something that I think featured into their analysis.
Great, thank you.
Yeah.
Aaron, Dr. Modiano, I think, has his hands raised, and then Dr. Pinnell after that.
Great, I will, Jaime, go ahead.
Hey, thank you, Susan. Really nice talk. So I'm going to play a little bit of devil's advocate and see what, try to get your reaction.
Okay.
And I've got maybe 3 points. The first one is you pounded into our heads the positive aspects of the canine model, right? And we all have to recognize that a dog is not a small human or a big human, so there's clearly some differences. You also said at one point that the dog is a better model than a mouse, and I would just throw out a caution and really encourage the audience to never put a judgmental value on any model, because every model has its advantages and its disadvantages. And so I think that people who work on mouse models could easily argue why the mouse model is better than the dog model in many respects. So just as a sort of note of caution that these are all complementary, none are perfect. And I know that when you go to study section, you do not want your dog grant to be reviewed by a mouse person who you tell that their model sucks. So, you know, the other thing is that, you know, there's a lot of emphasis placed on data that are statistically significant, but sort of biologically irrelevant. And Yoji might actually want to input in here. In the T-reg data that you showed us, they're showing asterisk data where the differences go from 0.9% to 1.3%. And I can tell you that as a clinical pathologist, I would not be able to categorize a patient of any species into a risk group in a difference between 0.9% and 1.3%. You know, and so just because those differences might be narrow, I think that we have to be very cautious about making a big to-do about them. And I see that repeatedly where people just, you know, p-hack into being able to get that magical 0.05 value. And I think we need to fight against that. The third one goes to the gene expression data. And I think, you know, there's some really interesting things there. But one of the things that Aaron's Harvard constantly reminds me is that, you know, when you're looking at data from 26,000 genes in terminable combinations, finding 10 things that are alike by random has a really, really high probability. And so I guess the question is, do you know, and I think this goes back to the question that Sarah asked earlier, do you know that these genes that are highly expressed in the human and dog tumors are really co-regulated by either a driver event or an organizational process, so that you really need those 10 genes to act in concert to create the tissue that we call oligodendrioma or GBM, or do you think that maybe this is really a statistical artifact and we need to really dig deeper into maybe using, you know, novel technologies like spatial genomics or other things that actually place those individual genes in the context of cells and organization? There's a fascinating paper in Cancer Research this week looking at differentiation of malignant glioma cells into perivascular cells that actually might contribute to that malformation of the blood vessel structures that you talked about today. And it would be really fascinating to actually be able to place the gene expression profile of those cells versus the meat of the tumor versus the other stroma. So I don't know what your thoughts are on that.
That all sounds, I think, wonderful. I agree with all of the things that you mentioned. I don't know specifically how much they confirmed in terms of if there are shared transcriptional drivers between some of these events. I'd have to just delve deeper into it to look into that.
Thanks.
Yeah.
Thank you, Jaime. Dr. Pinnell, Chris.
Hi.
Okay, can you hear me now?
Yes.
Thanks. So the CD200 receptor family has activating and inhibitory receptors, and the French Bulldog data are striking. So is one of the first things that you plan to do to look at the receptor family in French Bulldogs and see how they compare?
Yeah, absolutely. So what I probably should have stated, though, was that it's not just the dogs getting the CD200 for the French Bulldogs. It's across all the immunotherapy groups that they did poorly. But but they did do poorly in the CD200 group. So yes, that would absolutely be something that I want to investigate.
Okay, thank you.
Yeah.
Great. Thank you, Chris. There's another question here from Kiana. So, you did a wonderful job outlining similarities between canine and human gliomas, but what would you say are the really key differences that we need to be aware of as clinical trials kind of move forward?
Yeah, that's a great question. So some key differences would be one, that I think the IDH mutation is a big one, because that's something that clearly drives, is driving gliomas. And so I think that that's one potential key difference that we need to figure out. The other key differences that we see, one of them is just kind of the age of the patients. And so it's a little bit unclear, like, are these dogs at 7 to 8, are they the same age as a child, right? So that's because most people, like children, when they develop these gliomas, it's about that same age range, a little younger sometimes too, but 7 to 8 is one of them. Or are they like the older people, right? Where, because dogs, they age progressively faster than people. And so that would be another key difference. There are many others. I mean, and so even though there were, some of these similarities kind of in the top 10 upregulated genes in canine gliomas compared to human gliomas, it's not necessarily crossover. And so some of the things that are upregulated in human gliomas are not upregulated in canine gliomas at this point. So that would represent a pretty significant barrier if those upregulated genes are shown to be key drivers of gliomagenesis in people.
Okay, great. Dr. Bluhar has her hand raised. So Aaron, could you help us with that?
Are you there? Are you there?
Liz, you should be on, I think.
Yep, you should be good to talk.
Out of the Jackson was talking when they were looking at the canine, comparing canine tumors to the genomics to the human and the pediatric tumors. There was a difference in there in that all of the human tumors, whether they were adult or pediatric, were actually surgical biopsies, and all of the canine tumors were from postmortem samples. And I don't know whether or not that might make a difference, but that was one of the problems that they noted in that paper that may account for some of the differences that they didn't see or similarities that they didn't see. So more of just a statement rather than
Thank you.
a question.
Yeah. Thanks.
Okay, great, thanks. Okay, good. I don't see any other questions. Let's see. I think we are, yeah. If there are not any other questions, I think we are right around 10 a.m. So it seems like a good time to conclude. Dr. Arnold, thanks again so much for the talk and presentation today and the great update on what's going on. So thanks again for presenting today. Yep. Everyone have a good day. Thank you.
All right.
Bye.
Thank you both. Thanks.
Bye.